that starting, and that's okay. So let's get started again. Please welcome Bart, who give a talk on immunological view of fetal individuality.
Okay. Hello. So first of all, thank you so much for giving me an opportunity to come here and to give this presentation. It's I really appreciate it, and there were so many interesting presentations before me that it's really hard to beat it somehow. So the title of my presentation is Immunological View of Fetal Individuality. Let's see. Okay. So I would like to start with specifying how I understand the question of fetal individuality. So, of course, this question has significant legal and ethical implications. Well, this question is often understood as the problem of whether a pregnancy involves one or two entities. And from a biological point of view, I think this question poses a major paradox in a sense. So on the one hand, fetus can be considered as a separate entity. It is a basis of a containment view of pregnancy, represented by authors such as Smith and Bogart. Now, on the other hand, fetus can be considered as an integral part of the gestating organism, a parthood view represented by Kingman. Well, intermediate perspectives have been also developed, and some authors suggested that a pregnant female cannot be considered as one or two entities, either because of the processual character of the pregnancy or due to the complex relationship between the mother and the fetus during pregnancy. Well, a special place in this debate is occupied by discussion of the role of the immune system. We were fortunate to attend the talk by John. Indeed, several authors have asked about the role of the immune system in defining individuality of the fetus. Well, so immunology is relevant to the debate because of its long-standing focus on self-non-self recognition, an idea that the immune system can distinguish between what belongs to the organism and what does not belong to the organism. And also, it is relevant to this debate because of its long-standing focus, like on the question of, or its potential to reject foreign tissues. So that's another aspect of it. So as with the more general biological consideration, immunology-based arguments have been used to support a spectrum of approaches. So at one extreme, there is a foreign fetus model, which was developed by Medawar, a model that, according to which the fetus is recognized as a non-self entity. And this perspective aligns with the fetus as a separate entity model. Okay, so developed by Medawar, as I said. So he kind of conceived a fetus as a kind of semi-allograft, as he called it, semi-allograft, a foreign transplant, which is not rejected. He said it's like a transplant, but it's not rejected. Why it's not rejected? Because he thought it's most likely because it is completely sequestrated from the maternal immune system. Very well, I think, aligns with the containment view. On the other hand of the spectrum, there is the view of the immune system as supporting the kind of integration of the fetus with the body of the gestating organism. And this was considered by Kingman, who noted that the maternal immune system of the mother encompasses or shields not only the gestating organism, but also the fetus. And this is a clear case for the parthood model of pregnancy. And so intermediate approaches have been also developed And immunology was used to support a relational view of the fetus and gestating organism, where both entities are assumed to maintain a certain degree of distinctiveness while being intimately connected to various immunological interactions and adaptations. So Fiona Hoes, for example, she was developing this kind of neither one nor two entities model based on the relationship, like very intimate exchanges between the fetus and the mother, mediated by the immune system. Okay. So as you can see, there is a whole spectrum of approaches. And now recently, in a philosophical context, Arjun Davensan suggested that the maternal immune system recognizes fetal cells as essentially foreign somehow, even though promoting tolerance after all. So he didn't commit to a defined view of fetal individuality, although like in his last presentation, we could hear it. So I was not sure quite how to place his perspective. Fetus as non-self, but tolerated non-self, if it makes sense. So I was not sure how to position it, but I think based on the presentation, it would be much easier. Okay. So thus at the end, it's not clear whether the maternal immune system recognizes the fetus as self or non-self, or how this recognition relates to the question of individuality in pregnancy. Self and non-self. What is really self and non-self? Because it's at the center of this kind of debates. So critical for developing the idea of self and non-self discrimination were experiments by an American immunologist named Ray Owen. So he observed the following. If there are two bulls with different blood types, one having SX2 blood type and the other RL blood type, and they both breed a cow, inseminate a cow, and then this cow carries two twin calves, here labeled as D and E, that share placental circulation during development, then what happens is that these two twin calves, despite having different genetic fathers and therefore different blood types, or despite having like originating, having genetically different fathers, they have blood types of these two bulls. So each calf carries blood types like SX2 and RL2. Have the same blood. They have blood types from both of them because of the shared blood circulation. Because the blood circulation is shared between them, therefore they have the same blood type. And yet their immune system, even though they are not genetically, they don't have this one of these blood type encoded in their genes, they're tolerant to this other blood type. I hope it's clear. So this condition is known as naturally acquired chimerism. And it showed that the immune system could learn to tolerate foreign cells if exposed to them during early development. So what this experiment shows is that, and the same in humans, if you're exposed to an antigen early in development, then you become tolerant of this antigen during your lifetime. This is an interesting, remarkable observation. Now, to make sense of this observation, Macfarlane Burnett, he developed a model, a kind of a scientific account of what's really happening in these cattle, or it may be other mammals. Namely, he suggested that the embryonic stage represents a very unique period in the development of the immune system. Okay, this is what's happening. So if during this stage, an immature lymphocyte of an embryo encounters its specific antigen, regardless of its genetic origin, it will become clonally deleted or eliminated from the lymphocyte repertoire. And these encountered antigens become defined in this process as self for the immune system. So this is what self is. So self is not something that is genetically encoded. It's something that, like, our immune system learns during embryonic development. It's not, you know, it's not in our genes. It's about how the immune system learns. And that's an important observation. And in the adulthood, the lymphocytes that encounter an antigen, they are non-self if they have not been previously encountered by the immune system. They will become activated and they will expand to reject or to protect us or to reject these self-antigens. So, yeah, this is the self-non-self model. So the details aside, I mean, this model has been modified massively and became much more complex than I presented. But one conclusion is that genetic self and immune self are not the same. So if we are talking about fetal individuality or other individuality from an immunological perspective, we are not talking about genetic individuality. Because from the genetic point of view, we can say the fetus and mother are different, right? These are distinct individuals from the genetic point of view. But now this, again, brings us to the question, are they also different immunological individuals? Burnett said the recognition of self is something learned during embryonic life and not genetically ingrained. And this is the whole idea of self and self-discrimination. So now, so having introduced the idea of immune self and emphasizing the matter of learning, or, you know, it's not something in the genes. Now we should ask ourselves the following question. Are fetal cells immunologically foreign to the mother? Immunologically foreign, genetically they are, but are they also immunologically foreign? Now, to answer this question, we should note that there are like basically two types of fetal cells that the immune system of the mother interacts with. One type of cells are extravillous trophoblast cells, these EVT. These cells originate from cytotrophoblast and invade into the maternal uterine tissues during placental development. Okay, this is the fetal side, and this is the maternal side. And these are the extravillous trophoblast cells. So are these cells immunologically foreign to the mother or not? That's the question. So the answer pretty much depends on whether these cells express foreign HLA molecules and how they are recognized by the immune system. What are HLA molecules? Arjun mentioned these molecules in his presentation, in his paper. So HLA molecules, these are the types of molecules that must be matched between the donor and the recipient in, for example, organ graft procedure. I mean, if you have an organ transplantation, there must be a match between your HLA molecules and the donor HLA molecules. You know, right. So now the question is: do extravillous trophoblast cells, do they have these foreign HLA molecules and how they are recognized? So do these cells express these foreign markers? Well, it turns out that they do not express the classical HLA molecules, such as HLA-A, like most other tissues in your body would have HLA. They don't have a HLA-B, which is another kind of marker of, genetic distinctiveness. They don't have HLA-B. The absence of these molecules is a major adaptation for the maternal immune system to tolerate these cells. Yes. Instead, the extravillous trophoblast cells, they have certain non-classical HLA molecules, such as LHA-E, LHA-E, LHA-G, that have, all of them have a very much tolerogenic function. All of them basically promote tolerance to these fetal cells, extravillous trophoblast cells. The only classical HLA that is expressed on these trophoblast cells are HLA-C, which despite being able to present paternal antigens and thus being genetically foreign, so they are genetically foreign to the mother, these HLA-C do not trigger rejection. They do not trigger rejection. Instead, what they do, they activate NK cells, which mediate tissue remodeling to promote successful placental integration with the mother. So what does it mean? Yeah, adaptation to promote. So the conclusion is the maternal immune system tolerates these extravillous trophoblast cells rather than recognizing them as foreign. So the immune system of the mother sees them essentially as their own, as a mother's own cells, does not reject them. It tolerates them, actively actually tolerates these cells. So let's look at the other class of fetal cells the maternal immune system interacts with. And these are microchimeric cells. I will draw conclusions about the individuality, of course, at the end. I mean, this is what we are going. So what about microchimeric cells? So the cells, we know that cells from the fetus enter maternal circulation and persist for decades in various organs, tissues, and the circulatory system of the mother. This is a phenomenon of fetal microchimerism. Now, at the same time, maternal cells cross the placenta, enter the fetus, and persist in the fetus. Maternal cells persist in the fetus after birth. And it was counted that 1 in 1,000 peripheral blood mononuclear cells, 1 in 1,000 come from the fetus in the mother, 14 weeks after of gestation, not after, during gestation. So this number is growing. This number of the fetal cells is growing, and it's quite significant. So never mind the technicalities here, but what I would like to tell you is that the microchimeric cells from the fetus are genetically foreign to the mother. Are they? Yes, they are. They express HLA molecules of the father that could otherwise trigger rejection. However, in the context of pregnancy, these cells are actively tolerated. How? For example, there are cytotoxic CD8 T cells. They normally mediate organ graft rejection if you have an organ transplantation. And these cells in the mother are clonally deleted. They're eliminated. There are studies to demonstrate that. Also, CD4 T cells, another class of T cells, it's not, this is not that important, but that are specific for the genetically foreign peptides, differentiate into specialized immunosuppressive cells. These are immunosuppressive cells that are called regulatory T cells. And just to promote tolerance, actively promote tolerance towards these fetal cells in the mother's body. So insofar as the fetal cells are tolerated and incorporated as mother's own, so they become eventually these immune cells, despite genetically foreign, they are tolerated by the mother as her own. They're incorporated into her system. They persist in the system. There are even studies showing that these cells, they they help to repair the tissues in the mother's body. They may play many different roles, but they become part of her, constitution in some sense. So they, and this is, I think, the key word, they become part of her extended immune individuality that goes beyond her genetic constitution. So her immune individuality expands, literally expands during pregnancy and incorporates these fetal cells. At the same time, the maternal cells are tolerated in the fetus, created an extended form of immune individuality in both directions, you can say. So there is this, so this was noted and in the literature, and there is, for example, there's this paper about the changing maternal self hypothesis. So a paper from 2007, and the authors, they say, look, maternal self expands, changes during pregnancy. And the same happens with the fetal self. It's also expansive. Right. Yeah, so I mentioned that the same happens with the maternal cells. So now what to make out of these observations? So how can we construe a maternal fetal relationship in this immunological framework? So we have parenthood model that I mentioned, the containment model that portrays the fetus as potentially hostile immunologically, and therefore requiring some kind of separation. There is also the not one but not two model. It's like kind of shades of gray here. We don't know exactly. And then there is the, I don't know how to, is it an overlap? It's a kind of merged overlapping individualities model. So they overlap, but they match. They kind of maybe become part of each other. It brings us closer to kingdom. Okay, so I think that this view of fetal individuality, supports this kind of view of individuality. I mean, in a sense, I mean, people specializing in metaphysics could help to conceptualize it, but the way I see it is that basically the fetal cells, they become part of the maternal immune individuality, which expands. So there is a kind of maybe part-whole relationship after all. But this is not the end of the story. There is something that maybe will surprise you about this chimeric individuality. To understand how the immune individualities of the mother and the fetus change during pregnancy, consider that the microchimeric cells can be transferred beyond the direct mother-fetus pair. Now, these cells can be passed between family members across multiple generations. So, for example, if this is mother and this is her daughter, the daughter has now some of the maternal cells in her body, and then she may pass some of the cells, these cells that are not her own, they are not genetically her own, but she can pass these cells maybe from her grandmother or great-grandmother to her daughter. You see what's happening? This is a very complicated image. So this is how it has been represented. So, for example, let's say you have an older brother. Now, this brother seeded his cells to the mother's body, and then the mother seeded these fraternal cells to you. Like this is this kind of situation. And then from generation to generation, some of these cells may be transferred to other, and they are detectable. There are studies, and you can find these are whole cells. These are not, because you can also find fetal DNA in mother's body, but you can see also the whole cells, the persistent stem cells that are there, and they can differentiate into different kinds of cells, and yet they are genetically foreign to you because, for example, they have Y chromosome and you have XX, for example, or the other way around. So, right, so how to look at the fetal individuality? I mean, how the fetal maternal individuality from the immunological perspective. So we consider this kind of situation in which there is a kind of expanded immune individuality encompassing the mother's body, the fetal body together in a sense. But now considering that there is a network of tolerance and expanded tolerance, it's much more complex. It may also, this immune individuality may therefore encompass other genetic organisms like here or here. Or there may be also such a form of immune individuality. And this. So, well, so immune individuality, or this also may be like one form of individuality. It can cross the time, generations, encompass multiple individuals. Yeah. So genetically distinct cells are mutually tolerated and recognized across space and time, creating an extended immune cell, extended immune individuality, we would say, that transcends both physical boundaries and temporal limitations. Okay, so conclusions. Well, the maternal-fetal immune relationship demonstrates that, first of all, neither the mother nor the fetus operate as self-contained autonomous individual during pregnancy. The maternal-fetal interface creates a shared immunological space of tolerance that may encompass multiple organisms across time and space. These dynamically interacting and overlapping spaces generate emergent configurations of immune individualities in a sense. These individualities are, they may span across generations, they may, yeah. So thank you so much for your attention.
Questions? Let's start with RJ, and keep your hands up and I'll let you down.
Thank you very much. That was interesting. I don't disagree with any of it, but I feel like you push the individuality notion too far, right? So it is true that fetal cells cross into the maternal circulation and reside there long after pregnancy is complete, and those cells are not rejected. But that does not mean that the immune self of the pregnant, now no longer pregnant organism will tolerate a transplant from their daughter, right? So a daughter cannot give her kidney to her mother without the mother taking immune suppression. So there is a limit on what kind of tolerance we're talking about. Equally, in the case of acquired immune tolerance, in the Owen's case, right?
Yeah.
Subsequent experiments show that if you introduce a fetus to a bacterium, that it will not then grow up to tolerate said bacterium or virus. This engenders sensitization. So the fetus will then have an augmented response to the bacteria or virus. So acquired immune tolerance in the embryonic stage is not, ubiquitous across all molecular markers.
Yeah.
And that's because genes do matter to immune tolerance, right? So there's, there are genetically encoded receptors, which, natural killer cells mean that decidual natural killer cells will tolerate MHC molecules from fosters, but peripheral blood natural killer cells will not. Immune cells in the gut will tolerate E. coli, but immune cells in your pancreas will not. Right? And all of these are genetically encoded into the receptor morphologies. So yes, microchimerism is cool and interesting, but how exactly you can go from we have a few cells that cross generations to this is all one conglomerate immunological individual, I don't see how that follows.
So yeah, so I think it's an excellent question. Exactly. I mean, I agree that there are limits, of course. I mean, it's not, but there are limits. But also, these limits show us something important about the nature of immune individuality. That it's a matter of degree. So it may be, for example, we don't have to do with well delineated, robust individualities, as we would like to see, like carved in the stone, in a sense, and but it's a matter of degree. So let's consider this case of microchimerism. Yes, for example, if you have your maternal cells, okay, let's say, yeah, you have your maternal cells in your body. You may still reject, your organism may still reject the graft from mother, but for example, your organism is more likely to accept the bone marrow transplantation as evidenced by the studies. Also, if, for example, your mother is Rh positive and you are Rh negative, then you are more tolerant to the Rh positive blood transfusion, for example, if it happens. This was also shown. And there are also other numerous studies that I cannot, cite by heart, but I can send you references to, just to show that, yes, there is a degree of, I don't know, of rejection, but there is also a certain degree of tolerance that you acquire because of these microchimeric cells that you have in your body. So there are many studies on that. So this tolerance shows, and also there are studies showing that the changes that come, but if we just limit ourselves not to microchimerism and across generations, which may be a kind of a weaker form of immune individuality, but pregnancy itself, there are systemic changes in the immune system of the mother, in a sense that, for example, if the mother was suffering from autoimmune diseases, then these autoimmune diseases, it's very well known, become attenuated because many individuals have autoimmune diseases, and they see that during pregnancy they have a complete improvement. Why is it so? Because of this kind of reshuffling of the individuality of your immune system, which now becomes more tolerant to fetal cells and so on. It has to really reorganize itself. So these changes in immune individuality are most pronounced, of course, during pregnancy, and then they may fade away a little bit across generations, but they stay with you and they define, with all this intersecting, immune individualities that we participate with, it kind of also redefines what our immune systems are tolerogenic or immunogenic to. So Yeah, so I think this is an important point, and I do recognize that there may be shades, gradations of individuality, and this may not be as clear cut as in the classical logic that individual non-individual. This is a different kind of metaphysics here, overlapping individualities across generations that may fade away with time, and but also a subject of very intense research nowadays, actually. So the new studies are coming exactly as we are speaking. There's new studies on microchimerism and the immune system.
Yeah, thanks for the talk. I just wanted to ask about yeah, so you had the slide where you had the 4 images. You had the parthood and then either 1 or 2 thing, and then containment, and then your preferred way of kind of conceptualizing it. So it's just the gray dots that are in the mother. So okay, there are these cells that come from the fetus and they end up in the maternal organism. So I mean, there's a conceptual difference, right, between something that was part of something now being part of another, and something that is part of something also being part of another.
Yeah.
So why is it, I think it's for that representation to be kind of appropriate, the cells that have now made their way into the maternal organism need to still count as cells of the fetus in the sense of they're part of the fetus now, still, rather than just that they came from it in order to license a claim. I mean, yeah, I'm not sure exactly how you want to describe this in the way that you are, but however we want to describe it, that you would need that. So my question is, why should we think that they do still count also as parts of the fetus rather than merely having come from it?
So first of all, I have to apologize because I mean, this is just like my efforts to represent this complex abstract concepts graphically. But the way I think about it a little bit is along like a part-whole relationships in a sense, and expanded individualities. But let's say the maternal antigens, they become in a sense the part of the expanded individuality of the mother in a sense. So how to represent this was a little bit difficult for me, but I think that you understand my point. My point is that yes, these cells, they were not before part of the mother's, let's say, individuality, but they became incorporated in the process of kind of active tolerance to expand the scope. And the figure may be a little bit deceiving, I have to admit, but there is some kind of shared space of tolerance, and I honestly do not think that there are any parts of the fetus that would not be tolerated by the maternal immune system, or vice versa. It's just that the most of the body of the fetus will be sequestrated, but then the different types of fetal cells would seed the mother's body, many classes, and they would be still tolerated, and vice versa. So it's not just one type of cell. There are many of those, and they are all of them equally tolerated, and all of them express the whole repertoire of MHC molecules, these HLA molecules that are markers of, let's say, genetic individuality, but not necessarily, as we see, immunological individuality. So yeah, how to put it into, but this also reflects a little bit my coping with the question of like how to actually metaphysically, to frame it.
Thanks so much. Yeah, really liked your talk, and I think it's an interesting approach. But so my question was very similar to what Alex brought up, and I'm not entirely sure. Yeah, I'm gonna ask again because I'm still not entirely sure if I've followed your answer. But so if we assume that genetic identity / genetic individuality is not immune identity / individuality, how can we get from essentially cell swapping these cells that hang around in the body for years or decades to extended immune individuality? Because surely yes, they're genetically non-self, but genetic individuality is not immune individuality.
Right.
So is it just a case of, oh, this is because it is genetically non-self, we would ordinarily expect an immune reaction and there isn't one?
Yeah.
Or is it something else? Because I mean, lots of things can, LSD can hang around in the body for your entire life if you, you know,
And it will not be.
and we didn't, you know, we wouldn't consider that to be, I mean, I'm not saying that microchimerism is the same as LSD, but you know what I mean? Things hanging around in the body. I'm also not convinced that this forms an extended immune self in some way.
So this is an important problem. So here how it works. I mean, the truth is that, you know, when we think about like self and non-self, it's in the context of us like individuals. So we are thinking about it like biological individuals. So one biological individual will reject a graft or a transplant from another individual consistently, unless he or she is immunosuppressed. So if you have use a very powerful immunosuppressive drugs, then you may accept the, you know, the donors. So we are talking here about like the organic systems, like not chemicals or with which the immune system may not directly interact with, or, you know, and then there are these markers of genetic individuality on the surface of these cells. And these markers of genetic individuality, they are called HLA, human leukocyte antigen. So they have to be matched for the best result of the organ transplantation. And yet in the pregnancy, we see something very weird, that despite the cells expressing these markers of genetic individuality, so genetically they're undeniably non-self, but yet the immune system induces actively tolerance towards them. How does the immune system know that these foreign cells should not be rejected, but rather incorporated into the body of the mothers? I think it's the holy grail of, like, you know, reproductive immunology nowadays. So if this is the question, then of course I can tell you that this is a very important one. But we only know the mechanism. So we don't know how it happens. We don't know the first steps. Like, for example, you have the foreign cell, genetically foreign cell of the fetus. If you had a foreign cell, let's say from another person, from not a fetus, but for somebody else, it would be most likely very quickly rejected by the immune system. And here it is actually incorporated. It plants itself in a particular organs. They were found in the brain of the mother, like decades after she gave birth to the child. They were found in the liver, in other organs, and also in her circulation. So even though this number of these cells diminishes over time, they are still there and they are not rejected by the immune system. But what are the first steps? How does the immune system know to tolerate these fetal cells and not, for example, other foreign cells of, for example, of another individual? This is, I think, this is the subject of a major kind of research. So this is a kind of let's say big question, I think, of biology, you know, and I think this can be also only already answered empirically, because I mean we can speculate on that, but how it happens, how the immune system knows that, oh, this is the cell that I should tolerate. Probably it's related to, like, you know, the early stages of pregnancy that send some signals, and they, you know, they kind of instruct, in a sense, these T cells to either commit suicide if they see a fetal cell, or to become fully immunosuppressive afterwards. So, yeah. Thank you. Important, very, very key question.
Thanks. So yeah, so on exactly this empirical speculation, I was wondering, do you know if there is any research on whether microchimerism occurs to the same extent or at all in the case of pregnancies using donor ovums? Because if not, then would that suggest that maybe there is not just a self, non-self distinction, but like a semi-self perception by which the body can recognize, oh, this is at least partly genetically me?
No, I mean, it would take place during every pregnancy in all, like, many organisms, also not only in mammals, but you know, mice, and you know, so yeah, I think any pregnancy would involve this kind of transfer of bidirectional transfer of cells from the mother to the fetus and the other way around. yeah, regardless.
Elsie?
Yeah, thank you very much, Bart. That was great. So I have I'm just going to once more, I think, have a shot at articulating the question that Alex and Teresa asked, and I'm also going to say something else. Is that allowed? Okay. So, by the kind of merge, you distinguish very clearly between an immunological individual and a genetic individual. But then the merged picture you give, it's a fascinating story. I find it's quite comforting. And I'm like, oh, my mother's cells will always be with me. Not so comforting that my brother's cells might always be with me. But that story about emerging across generations, that of course is the genetic individual, right? So maybe my grandmother, who's long dead, her in terms of the genetic individual that my grandmother was, may well persist in me and my children. But because you yourself draw this firm distinction between the genetic individual and the immunological individual correctly, I think that the immunological individual would extend that way.
Yeah.
It's quite a different claim. And it'd be interesting to figure out what the arguments for that claim would be, because they can't appeal to the genetics, obviously, because that's the genetic case. The other thing I was wondering about was, so I don't know, I can't even remember whether I have or haven't actually published this, but I think if you look carefully at Ellen Clark's account of individuality, one thing that comes out of there is that, what starts as a question that is highly dichotomous, right? What are the individuals? And we expect to find objects that are individuals and objects that don't count as individuals. I think she merges, the question changes throughout her work, maybe possibly imperceptibly, such that individuality becomes this property. And it's a property you can have by degrees, right? So you can be like more or less hairy. It's not just that there are hairy people and not hairy people. You can be more or less as an individual. And I don't know if I've written that down yet. It's on my computer. But I think I've written down somewhere suggestively that maybe reproduction in all of us, of necessity, kind of requires that we slightly downgrade our individuality and coherence, right? So just that we can be temporarily less hairy, theoretically. You temporarily accept a lesser degree of individuality in order to facilitate, making another organism. And of course, mammals, female mammals, have taken that to a fair amount of extremes. And I think possibly that kind of view merges really well with this very nuanced story you tell of the immunological story, right? Because I think one thing that comes out really nicely from your view is that the story of the immunology is not one that is like, it's accept or reject, but it's like, there are all these parts of the immune system, and some will do this, and some will do that, and some will do that, and so on. And arguably, the overall picture is one of, not a disappearance of individuality, not emerging, but certainly a downgrading. So that might be a helpful type of view to use in conjunction with you. Would also be interesting because Clark isn't really into immunology, and her big enemy is Prideaux, who thinks, anyway, that's a further story we can talk about. But anyway, that wasn't a question, was it? Oh no! Oh no! Do you think that might help?
I appreciate this. These are questions, but so about the this fading individuality. I mean, we mentioned that also. That absolutely. So with you can have a different degrees of tolerance. I mean, the story, of course, is very complicated in the sense that it's not necessarily the way we think about it. You have a rejection or acceptance of something, and you have like a clearly cut individuality, like it was, the original self no self story was portrayed. But there may be, different degrees of responsiveness or tolerance. You can have like a very strong tolerance towards something or less of a tolerance. All of this makes a lot of sense, and also like helps to really put the idea of microchimerism or the immune individual chimerism into a proper perspective, that we can have like a different fading levels, and also maybe during the course of pregnancy itself, the also individuality will become more and less depending on the stage and so on. So this is absolutely very, very helpful. And And your first comment about the genetic versus immunological tolerance, individuality. So indeed, I mean, I think they should be distinguished because, I mean, genetic individuality is very easy to spot, in a sense, to test. But with immune individuality here also, I mean, I didn't manage to explicate all the definitions or the assumptions, but the assumption is here that, okay, that if you have a genetic individual, immunological individual, depending on what it tolerates, like, okay, if it, you know, whatever it tolerates, your body tolerates it, it's part of your immune individuality in a sense. So it's a very simple definition, but I think that's also Arjun was also referring to this kind of. So if, for example, my body tolerates, like my own cells of a fetus, then the cells of this fetus become a kind of, and they become incorporated into my body. They are part of my immune individuality. It's also not only being tolerant of these cells, but also like fostering their survival, and you know, it's also immune-mediated process. So yeah, so this will be different, and just to be precise about that. And I'm afraid that maybe efforts to define individuality outside of this kind of assumptions. So for example, can we just talk about biological individual in general, outside of the context of genetic individuality, physiological individuality, immunological individuality, maybe very difficult. I'm not saying impossible, but.
So in a way, I think this comes back to the same issue, but really, so it was a finger really on Elsalin and Alex and Teresa. I just wondered what you might say about cases, so immune responses to cancer. Because that seems to be a case. I take it other people know more about this, but I always took it that Prideaux would be very comfortable with saying that cells which were from the original germline were not part of the organism on the basis that they were attacked in those kinds of pathological cases. So that seems to be a kind of your diagram where you had the sort of the lots of little dots. Presumably, are there some black holes if there are cells from the original germ, from the original zygote, which are no longer being tolerated?
Yeah, so about the immune individuality in cancer, I mean, of course, I mean, this is a very big kind of topic, but so there is just roughly, there is like the idea of immunosurveillance, that the immune system scans the body in search of altered cells, mutated cells, to eliminate them. And this could be actually the case of for example, if a cell, for example, misses this HLA molecules, it would be rejected by NK cells, for example. This is like part of the protection mechanism against cancer in the body. Or it displays some so-called neoantigens, it's called neoantigens by these molecules. So in that case, I mean, to be consistent with this model, we would say that these are distinct individualities in a sense, immunologically, because they are rejected. They are not only genetically different, but they also are recognized as null self, and they're eliminated. But what we see in cancer is that very often the immune system becomes tolerant of cancer. And what is really interesting is that the immunologists find the same mechanisms that are involved in the tolerization towards the fetus in the tolerization towards cancer, which is detrimental for us. But they see the same pattern in which a cell that is highly mutated, genetically foreign, becomes kind of tolerized and incorporated into the system. So there may be like a kind of shared mechanism of conversion or expansion of tolerance, expansion of immune individuality in different scenarios. And this would include not only pregnancy, but also cancer and maybe other forms of let's say, long-term tolerance. So the example of cancer would be just application of the same logic to a kind of pathological case, and believe me or not, but the same kind of cells that polarize the mother towards the fetus are involved in this process. So that's why it's also interesting.
And kind of susceptibility of species to cancer. So are we more susceptible than organisms that say don't do placental pregnancies at all, or is there variation?
Maybe we're going to have to wrap up now, but maybe
it's a
you can talk
fascinating
about this
hypothesis.
in the.
It's a very, very interesting hypothesis.
Cool. Okay, great. That was brilliant. So we're going to take a break now. We'll be back at midday.